We're glad you've joined us for this live webinar, Understanding Immunosuppressive Myeloid Cells and Immunotherapy Using the Proteome Profiler Antibody Array. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Bio-Techne. Bio-Techne Corporation is a leading developer and manufacturer of high-quality purified proteins, antibodies, immunoassays, biologically active small molecule compounds, and advanced cell diagnostics in situ hybridization detection products. Bio-Techne's products are integral components of scientific investigations into biological processes and the nature and progress of specific diseases. To learn more about Bio-Techne, please visit www.biotechne.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE tab at the top right and follow the process to receive your credits. You can pose questions to the speaker during the presentation while they're fresh in your mind. Please note that Bio-Techne scientists Rachel Weller-Roska and Anna Vervoort are available to answer questions about the proteome profiler antibody arrays used in this webinar. Simply type your questions into the Q&A box. Questions will be answered after the presentation. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window or report your problem through the Q&A box. I now present today's speaker, Xin Lu, PhD, the John M. and Mary Jo Bowler Assistant Professor of Biology at the University of Notre Dame. Dr. Lu develops novel insights into the genetic and molecular mechanisms of prostate tumor progression, tumor immunology, and treatment resistance. His research team at the University of Notre Dame and Indiana University's Simon Cancer Center is actively investigating the mechanisms of myeloid cell regulation and targeting in prostate cancer and other forms of malignancy, including rare cancers. Through his PhD research at Princeton University and postdoctoral research at MD Anderson Cancer Center, Dr. Lu has made many contributions toward developing a mechanistic understanding and identifying therapeutic interventions for breast cancer and prostate cancer. He has published more than 20 research and review articles. His complete bio is found on the LabRoots website. In this webinar, Dr. Lu will discuss how he used antibody arrays to interrogate signaling changes associated with immunotherapy in animal models. Dr. Lu will now begin his presentation.
Hello, everybody. Thank you so much for the invitation and the introduction. Again, my name is Xin Lu. I'm an assistant professor in University of Notre Dame. Today, I will mainly talk about how to use the proteome profiler array to study cancer immunology and immunotherapy. So first, I'd like to quickly go over my laboratory at Notre Dame. Our research goal is to develop novel insights into the genetic, molecular, and cellular mechanisms of prostate cancer progression, including other cancer types, as well as understanding of the tumor microenvironment and the treatment resistance. By using those knowledge, we hope to develop and improve the therapeutic interventions, so eventually we can benefit the cancer patients. So our focus is on prostate cancer, breast cancer, but we also do a few rare cancers because our lab belongs to the Center of Rare and Neglected Disease at Notre Dame. So the current central project in the lab include understanding the cancer cell intrinsic and extrinsic mechanism for immunosuppression and immunotherapy resistance. We also focus on using genetic tools to understand the tumor progression and also develop new targeted therapies against the cancer. So my contact information is here. So if you're interested in further discussion or collaborations after the talk, please contact me. We'll definitely welcome all kinds of collaboration opportunities. So today I will mainly talk about prostate cancer. As we all know, prostate cancer in the Western world is the most frequently diagnosed malignancy for American men and also men in Europe. So as shown here, about 19% of new cancer incidence is the prostate cancer, and also it's the third leading cause of cancer mortality for American men. Over the years, there have been a lot of studies trying to understand the mechanisms of prostate cancer progression. So now we understand that prostate cancer can derive from the normal prostate gland, and then go through some histologically defined processes such as the early prostatic intraepithelial neoplasia to adenocarcinoma, and then eventually become metastatic adenocarcinoma. And then during this process, there has been characteristic genetic and epigenetic changes associated with the steps. For instance, the PTEN inactivation and EZH2 overexpression. However, despite this understanding, the current strategies to target prostate cancer, particularly the malignant metastatic castration-resistant prostate cancer, is very limited. So in this disease, the CRPC remains incurable currently. So over the years, the field gaining a lot of knowledge about tumor microenvironment and how it plays a role to regulate the different hallmarks of cancer. So this diagram is the famous cancer hallmarks that's initially proposed by Dr. Weinberg and Hanahan, and then recently revised to include additional hallmarks. And then if you look carefully, and then you actually see that the non-cancer cells in the tumor microenvironment, which include the infiltrating immune cells, the fibroblasts, and then the vascular cells, can regulate many of these hallmarks. So they are very increasingly be appreciated to play important roles and also modulate the response to therapeutics. And today we mainly talk about this particular population called myeloid-derived suppressor cells, or MDSCs. So myeloid-derived suppressor cells is a heterotypic population of myeloid cells that are induced by cancer or other diseases such as acute infection. So this diagram shows both the normal and abnormal differentiation pathways for myeloid cells. The thin dotted lines here represent the normal differentiation. As you can see that these cells come from the bone marrow hematopoietic stem cells originally and become the progenitor cells. And those progenitor cells can develop into monocytes, which become macrophages after enter to resident tissues. They also can develop differentiate into granulocytes, including neutrophils, basophils, and eosinophils. They also can differentiate into dendritic cells. Those are normal health cells in the healthy individuals. However, in patients with cancer, the cytokines produced by either cancer cells or other stromal cells in the microenvironment can induce the abnormal differentiation pathways, and a lot of those progenitor cells actually are held in such immature status, and then display as MDSCs. Here it shows that the MDSC can at least have two subpopulations. One is called the polymorphonuclear MDSC, also known as granulocytic MDSC, because their morphology is similar to the granulocytes. And another group of MDSC is called the monocytic MDSC because their morphology is more look like a monocyte. So both of these MDSC population can inhibit T cell activity through different kinds of mechanisms. So today, my talk will focus on understanding the function of MDSCs in prostate cancer and how we can target them, and particularly highlighting the technique of the proteomic profiler array. So my research on prostate cancer and MDSC came from our earlier publication in my postdoc lab, Dr. Ron DePino's lab at MD Anderson. The lab published a paper in 2011 to show that we developed a very aggressive prostate cancer model by prostate-specific deletion of two tumor suppressor genes, the PTEN and the SMAD4. So you can see that compared to the PTEN single deletion, this double knockout develop aggressive prostate cancer very quickly, and those cancer also metastasis to lymph node and to lung. And then by looking at the histology of these tumors, of these double knockout tumors, you quickly realize they not only have a significant increase in tumor cancer cells, they also have a lot of infiltration of non-cancer cells, and a lot of them look like immune cells. That was the earlier paper. And then subsequently, collaborating with another postdoc in the lab, Dr. Wang, we profiled the tumors at different ages for immunocytes. And we see that on the right, you see that this red colored pie chart represents the CD11B positive, GR1 positive MDSC populations. They increase a lot over the time of the mouse progression. And then the flow cytometry also shows the population of MDSC in this mouse model are mainly dominated by the polymorphonuclear MDSC. This is true across different lobes of the prostate, the AP, the DLP, and the VP, shown at the bottom. So those are phenotypical data. And then I wouldn't go into details of this paper because it was published more than 1 year ago. So, but basically in this paper, we demonstrate that the prostate cancer cells will activate YAP transcription factor, and YAP and TEAD interaction will drive the transcription activation of the chemokine called CXCL5, and CXCL5 secreted from cancer cells will recruit the progenitor cells, the myeloid progenitor cells, which express the receptor called CXCR2 from the bone marrow, and those cells will migrate into the tumor parenchyma and then become those functionally immunosuppressive cells known as MDSCs. Those MDSCs will suppress the T cells in the tumor and then promote tumor progression. So if we treat the mice with some agents that can block MDSC recruitment or activation, such as CXCR2 inhibitor, blocking the receptor, we were able to partially inhibit the tumor progression in this model. That was the paper published in Cancer Discovery. However, as I said, we can only partially impair the tumor progression in this model. We cannot totally eradicate this tumor, right? However, this study did bring us to the field of cancer immunotherapy, which we all know it become very, very exciting recently due to a lot of clinical advances of new immunotherapeutics, particularly the immune checkpoint blockers, known as ICBs. So those ICB agents, such as anti-CTLA4 and anti-PD1 or anti-PD-L1 antibodies, are very important to reactivate the cytotoxic T cells in the tumor microenvironment, so that those T cells become reinvigorated and then display their anti-cancer activity. So here are the canonical understanding of how this antibody work. However, the metastatic CRPC, the castration-resistant prostate cancer, showed overwhelmingly de novo resistance to immune checkpoint blockade, shown by several publications. So the question is, why mCRPC is resistant, and also how can we develop new strategies to resensitize those cancer types so they can potentially be applied with the ICB reagents. So this graph shows the current treatment strategy for prostate cancer, from early local treatment such as surgery and radiation therapy, and also a lot of patients go through this hormone therapy, or known as antigen deprivation therapy, or ADT. But inevitably, these patients that gone through ADT will develop resistance and develop a CRPC. And when the CRPC emerges, the tumor can be treated with chemotherapy, such as some taxane-based therapies, but they only have very short-term benefit for the tumor, and eventually a lot of patients die from the CRPC disease. So our hypothesis is that at the stage of the emergence of the CRPC, if we apply a combination therapy of targeted drug that can block MDSC activity, but at the same time preserve the T cell activity, plus an immunotherapy approach, potentially we can reach some level of synergistic efficacy and then eliminate the tumor, or at least control the tumor in a state that it doesn't jeopardize the life of the patient. So that's the basic hypothesis based on our earlier studies. To test this idea, we need a very robust mouse model that's immuno-intact and also amicable to preclinical trials. So we chose this model, the triple knockout model that the lab recently published in 2012. This is a triple knockout model of deletion of P10, p53, and SMAD4 in the prostate of the mice. So we chose this model because this model also developed very aggressive prostate cancer, adenocarcinoma, metastasis to lung, metastasis to lymph node, and also occasionally metastasis to bone. And also because of p53 deletion, this model potentially harbor more somatic mutations that can potentially agitate adaptive immunity. So that's the basic idea of why we chose this model. And then to help us using the model, we actually incorporate some reporter alleles into the model. Here, particularly, we bred the mice with the MTMG allele, as well as the lock-stop-lock luciferase allele. So the MTMG allele allows only in the prostate cells are labeled with GIP, while the rest of the cells are tomato positive. Okay, and the luciferase allele will allow us to specifically monitor the progression of the prostate tumor using the non-invasive luminescence imaging. And also we optimized MRI imaging approach that we can use the data to quantify the tumor volume. So we optimize all these techniques with this so-called CRIP-small mouse model. But we also run into another problem, which is this mouse model based on this probation-Cree allele driver is not easy to breed because these males have prostate cancer. They have very low fecundity. So the problem is how can we overcome this so that we can have a large cohorts short period of time for preclinical tests. So we resort in this new technique called mouse embryonic stem cell chimeric modeling. So this is a very efficient preclinical technology that can create a lot of chimera mice that are ready for drug test. So basically the idea is we have this CRIP small mouse that have all those alleles that I showed in the last slide. And then we have those mice and we isolate a stable embryonic stem cells from those mice. And then we inject those stable embryonic stem cell line into blastocysts we isolate from wild-type mice, particularly the albino B6 mice. And then those chimera blastocysts will develop into chimera mice that have those incorporated embryonic stem cell lines. And remember, those cell lines were derived from those tumor-prone mice. So genetically, they are also making those mice prone to prostate tumor development. But because those prostates have both these cells derived from these cells as well as from the normal blastocysts, so that's why it's called a chimera. So, but this is the basic idea we try to accomplish, and then we did all the technical works to make this work, and eventually the lower table shows that by doing so we increase the efficiency of getting tumor-bearing males by 4-fold increase. That's just only talking about how many efficiency of getting the mice per cohort, like per liter. But we also save a lot of time. So by doing this, we were able to actually test our idea of combination therapy. So basically, because of this, result, we kind of compare to the old tech, not old, like traditional or conventional breeding-based gen model. We think this new mouse embryonic stem cell-based chimera model represents a new advance that we can further rely on for future preclinical tests as well. So now come back to the design. So as I said, we want to select drugs that can potentially target the MDSCs, but at the same time preserve T-cell activity. So we kind of search literature and also search for clinical trials, and we found 2 tyrosine kinase inhibitors. One is called dasatinib, one is called cabozantinib. We think are very interesting. So both of these TKI are FDA approved for treating other cancer types, but they were investigated extensively in prostate cancer and actually gone through phase 1, 2, 3 trials. However, they failed the phase 3 trial because they could not improve the overall survival of prostate cancer patients with CRPC. Nevertheless, these drugs proved to be active to regulate immune cells such as the T cells as well as MDSCs. So they have this immune modulatory activity. So we also, because PTEN deletion is very frequent in prostate cancer, we're also interested in testing a PI3 kinase mTOR inhibitor. We focus on this inhibitor called PEZ-235. So this drug is still in the early exploratory stage. However, one paper does show that this drug has low toxicity to T cells. So we think like this potentially can also be combined with immune checkpoint blocker. So we choose that as well. So we have 3 small compounds that we will test individually as a monotherapy or in combination with immune checkpoint blocker. And for immune checkpoint blockade, we use a combination of anti-PD1 and anti-CTLA4 antibody cocktail. That's our ICB treatment. So this slide shows the overall design of the preclinical trial. So by using the chimera modeling approach, we generate a whole bunch of these small chimeras, and then we set a criteria how to enroll them, which is the mice have to have their prostate over 150 cubic millimeter before they turn to 18 weeks old. Okay, so that's our criteria. If the chimeras meet that criteria, we will enroll them into our clinical trial. If they don't, we will not. They will be excluded from our trial. And this volume was measured by weekly MRI imaging. And once we confirm a mouse is eligible for our clinical trial, we castrate the mouse, and then we treat the mice with we feed the mice with the enzalutamide administered diet. So this combination of treatment of castration plus enzalutamide diet was optimized based on our preliminary data to show can induce CRPC in these mice, in these triple knockout mice. Okay, so after 3 weeks, we use MRI imaging to confirm the development of CRPC in this mouse, and then we randomize them into 4 groups, right? And then the mice will receive either the control, including vehicle plus IgG, isotype IgG, or the kinase inhibitor, or the anti-PD1, anti-CTLA4 antibody cocktail as ICB, or kinase inhibitor plus ICB. So the single treatment or combination treatment. And then the mice go through biweekly monitoring with MRI, and after 4 weeks, they will go to the endpoint analysis, which include tumor weight measurement as well as some histology and molecular analysis. So that's the basic flowchart of our experiments. So this graph shows the main data of this study. Okay, the most important data. You can see here there are 3 graphs here. On the left is the prostate weight. In the middle is the lymph node metastasis. On the right is the lung metastasis. So if you focus on the prostate tumor weight chart, the plot, you will see that neither the ICB or the drugs as cabozantinib or BEZ or dasatinib as single therapy, neither of those significantly change the tumor weight compared to the control. Okay, so neither of them is sufficient to modify the tumor. However, the combination of cabo plus ICB or the BEZ plus ICB were very effective to reduce the tumor weight to very low level. Okay, but this was not observed by the dasatinib plus ICB treatment. So that's the basic observation that we saw initially. That's very exciting to us. Okay, and this trend also held true for metastasis pretty much, except that in metastasis we do see that ICB alone have high moderate effect to reduce the metastasis burden but nevertheless, the combination of cabo plus ICB or the BZ plus ICB were the ones that really bring the metastasis burden down to almost zero. So this result was shown from the chimera model from one embryonic stem cell line, and in the paper we also show another cell line also have similar results. So it's not specific to a peculiar cell line. It's a general phenomenon in this mouse model. So these are some representative images of the tumor, including the primary prostate tumor histology, as well as GFE positive metastasis to lymph node and to lung. You can see that the most effective combination are the cabo plus ICB or the BEZ plus ICB. So to understand what caused the changes of the tumor by those immune cells, we use this technology called mass cytometry, or also known as CyTOF. So this technology will allow you to profile over 30 surface markers or intracellular markers at once without a need of compensation. So it's a great technology. So we use this one for our immunophenotyping, and immediately we saw that the dasatinib, chronic treatment with the dasatinib of the tumor of the mice caused almost a complete depletion of the intratumor T cells, including the total, both the subtypes, CD4 subset or CD8 subset of T cells. So this may explain why we don't see any effect for immunotherapy, combination immunotherapy, because you ablate T cells from tumor microenvironment, and now you trying to activate T cells. Of course, there's not going to be any effect. So this result actually retrospectively is consistent with the previous report showing the dasatinib inhibiting TCR mediated T cell proliferation in a different system. So in the following studies, we focus on the other two drugs that demonstrate a combination effect. So using CyTOF, we found that both the cabo and the BEZ were very effective to reduce the infiltration frequency of MDSCs, and also at the same time they also kind of boost up the CD8 positive T cells versus regulatory T cell ratio that's already increased by ICB, but increased further by this cabo or BEZ treatment. So this parameter, this CD8 over Treg ratio, was used frequently to indicate the effect, positive effect of ICB. So this was increased further by these two drugs. And then this result kind of suggests to us maybe the MDSCs compared to either cancer cells or T cells are more sensitive to cabo or BEZ. So to test this idea, we just use a cell viability assay to measure the viability of these three populations isolated from the same tumor to either cabo or BEZ. As you can see in the lower part, the blue line represents MDSCs. Indeed, those cells are most sensitive to the cabo or BEZ treatment. So that explains partly why they are the most pronounced impact population in the tumor microenvironment. So we know that this cabo and the BEZ can reduce the tumor MDSC infiltration, but what about their activity? So we isolated MDSCs from either the control-treated or the drug-treated mice, and then we co-culture those MDSCs with CD4 or CD8 positive T cells, and ask what's the consequence for the T cell proliferation. And you can see here, if you co-culture T cells with MDS isolated from control-treated mice, this MDS is very effective to reduce the proliferation of either T cell subset. However, if you isolate MDSs from the cabo or BEZ treated mice, those immunosuppressive activity was partly lost. That means that not only do these two drugs reduce the infiltration of MDSs, but also reduce the activity of the MDSs. In summary, cabozantinib and the BEZ significantly reduce the infiltration and activity of MDSCs. The question is, what is the mechanism of action for those drugs on MDSCs, right? So here comes the use of the proteome profiler array purchased from R&D systems. So the first we want to understand what's the effect of these drugs on the phosphoreceptor tyrosine kinase pathway, because we know cabozantinib targets some of the known RTKs. The BEZ is not directly targeting RTKs, but it can have some indirect consequences, presumably. So we just want to test what happens to tumors after the treatment using this protein array. So you see that in the 4 panels, we have control or ICB treated, or BEZ or cabo treated. Okay, and then you see that the ICB treated alone doesn't change much, whereas the BEZ or cabo treated tumors significantly downregulate those phosphotyrosine signals. And then we validate a few of those signals, but also expand our understanding by doing Western blot of additional markers in the RTK pathway as well as the PI3K pathway. On the lower right, you see that both the cabozantinib and the BEZ significantly reduce the signals for phospho-AKT, phospho-mTOR, the phospho-S6. Those are all components of the PI3K pathway. So this result is interesting to us because recently there have been a few reports showing that the PI3K pathway are important for myeloid cells to display immunosuppressive activity. So we kind of saw this again here in our data. So we want to validate the results by doing a staining for the MDSC markers, constant with a marker for PI3K downstream report, like a surrogate marker. For here we use the phospho-S6 as a marker for active PI3K signaling. As you can see here on the quantification result on the right, the percentage of MDSCs that are positive for phospho-S6 was reduced dramatically by either cabo or BEZ treatment. So this finding kind of consistent with our hypothesis that the cabo or BEZ compromise MDSC activity through inhibition of PI3K signaling. But these are correlative, so we need to provide some direct evidence for the idea. So we use this protein transfection method. So MDSCs are not easy to manipulate genetically, but we optimize the approach that we can use this protein transfection method to have some impact on the signaling of the MDSCs. So here we want to bypass the drug effect by directly transfect MDSCs with the P70S6K or ERK2. So by transfecting these two proteins on MDSCs, we hope that we can bypass the drug effects, which are more like upstream, and turn on the downstream pathways of the PI3K, right? And then once we do that, we see that the effect of cabozantinib or BEZ on the proliferation of MDSCs was relieved, right? So now you see that on the right, on the bar graph, you see that when you treat with cabozantinib, BEZ, there's a significant reduction of the proliferation activity and the viability of the MDSCs. But if you transfect the MDSCs with these two proteins, you partially rescue the viability of MDSCs despite the treatment of the drug. So that kind of suggests to us, indeed, the PI3K signaling is at least partially responsible for reducing the activity, the viability and MDSCs, and the activity of MDSCs in this model. So, and then next we want to understand. Okay, so now we know the drugs are hitting their targets. That's kind of expected. But MDSCs are known to be regulated dramatically by all kinds of cytokines. So do these drugs also have some effect on cytokine in the microenvironment? So to understand this, we again use the proteome profiler array from R&D systems. This time is the cytokine array. So we profile tons of cytokines using the tumor lysis from the control treated or the combination treated. And then we see that here we actually use the cytinib plus ICB as a control. As you can see here, the BEZ plus ICB or the cabo plus ICB treatment altered the level of cytokines, quite a few of cytokines, either decreasing them or increasing them. Okay, so a few of the cytokines that are decreased by the combination treatment are known to regulate MDSCs. For example, CTLA4, CD40, those are known to regulate MDSCs, positively regulate MDSCs. Now you reduce them, so it's consistent with the fact that those combinations reduce MDSC. On the other hand, the ones that are increased by the combination also have some known activity to regulate MDSCs. Here we particularly focus on this one cytokine called interleukin-1RA. This IL-1RA is a decoy for the IL-1 receptor, so it has no activity to antagonize IL-1 alpha or IL-1 beta activity to the receptor. So we designed this MDSC migration assay to show that MDSC are attracted by IL-1 alpha or IL-1 beta to migrate towards a source of tumor signals. But if you treat the system with IL-1RA, you can block such effect. But if you use antibodies that can neutralize IL-1RA, now the attraction of MDSC migration is recovered. So this result suggests that this combination can regulate quite a few cytokines, and in particularly can upregulate the 1RA, which antagonize the IL-1 mediated MDSC migration. So far the data suggests that those drugs have potentially direct effect on MDSCs, but as we now know, tumor microenvironment have all kinds of cells, including cancer cells. So very likely those drugs also have some indirect effect on MDSC through cancer cells. To test this idea, we designed this in vitro assay. We isolated prostate cancer cells from this triple knockout mice, right? And then we culture those cancer cells, and then treat those cancer cells with either vehicle or carbo or BEZ, and then we collect the condition media, and then wash off, use concentration and the reconstitution method to remove those drugs. And now those condition media are pre-treated with the drugs, and then use those condition media to culture MDSCs and see what happens to the MDSC. Did the pre-treatment with the drugs have any effect on MDSC activity? That's the basic question. So actually the first thing we saw is even you just use the condition media from cancer cells without any drug treatment, just condition media, you see that this condition media is very effective to turn on some of the key proteins, key genes expression in the MDSCs that are involved in like arginine depletion or reactive oxygen species production, like those NCF1, NCF4, or CYBB are all NADPH oxidase subunits proteins, and then those gene expression are turned on by condition media from cancer cells. So, however, if you use condition media from the pre-treated the cancer cells by the drug, now you see this level of increase was almost lost. Now the condition media lost the ability to turn on those immunosuppressive genes from MDSCs. And then we use the cytokine proteome profiler array to understand what cytokines are changed by those pre-treatment of drugs on cancer cells by compare those vehicle treat or carbo or BZ treated condition media. And we see that, again, quite a few of the cytokines are changed, not just single one, quite a few of them. Okay, so you see that those very well-known cytokines that regulate MDSCs, such as GM-CSF, GCSF, TNF-alpha, IL-6, those are all reduced by carbo or BZ treatment. And then this kind of brought us to the idea, okay, so now you are affecting many of those cytokines, so it's not just one of them. So to show that those cytokines can collectively affect the MDS activity, so we actually use the recombinant cytokines for all those changed cytokines, and then put it back into the condition media and show that now the expression of those important immunosuppressive genes are partly recovered when you treat the condition media, when you kind of supplement those condition media with the individual cytokines. So this gives us a conclusion that the drugs, the BZ and the carbo, not only affect the MDSC directly, but also affect what cytokines are secreted by cancer cells. By reducing the secretion of pro-MDSC cytokines, those drugs also reduce the ability of cancer cells to promote MDSCs in the microenvironment. So lastly, we were probed to use additional drugs to show that the PI3K isotype specific drugs also can collaborate with ICB to induce synergistic efficacy. So here we use two PI3K inhibitors, isotype specific inhibitors. One is the P110 delta selective inhibitor, one is the P110 beta selective inhibitor. Both, and also we tried a new drug called SX682. That's a CLCR1/2 inhibitor from this company. And then because we know that in these models, the CLCR1 and CLCR2 signaling is crucial for recruitment of MDSC to the microenvironment. So we, similarly to what I showed earlier, we either treat the CRPC with single drug of each of these, or ICB, or we do the combination treatment. You see that we see that the combination of the isoform specific inhibitor is very effective when you combine with ICB to elicit a significant anti-tumor effect for the CRPC. And this was also seen with the CLCR1/2 inhibitor in combination with ICB. So finally, we collect some clinical evidence to support that the granulocytic MDSC we have a lot in this tumor are also relevant in human prostate cancer, and they also inversely correlate with CD8 positive T cells. So these are data from fine needle biopsies, the fresh tumors that come from the clinic. So this inverse correlation between granulocytic MDSCs and the CD8 positive T cells in the clinical further corroborate the idea that we got from the mouse model, because in our mouse model we also have this inverse correlation. We also have the granulocytic MDSC as the dominating population of MDSCs. We also saw this in the clinical samples. So in summary, by using new mouse models and then a combination of ICB with kinase inhibitors, we show that for resistant tumors, for tumors that are resistant to immunotherapy, cancer immunotherapy, you need to have combinations of T cell activating drugs such as anti-PD1, anti-CTLA4, anti-PD1, but you also need reagents that can potently suppress MDSCs, such as the PI3K inhibitors or the CLCR2 inhibitors, in order to remove those negative effects on T cells. If you only do one of those, will not be effective. But if you do them together in a proper way, you will be able to elicit strong anti-cancer synergistic effect. So this paper was published earlier in 2017, and then now my lab is continuing on this direction to understand deeper how MDSCs regulate T cells. In particularly, we have been focusing on how MDSCs, by producing ROS and RNS, to modify T cells, like very important proteins on T cells, so that they can have new mechanism. We identify new mechanism how MDSC regulates the T cells. So finally, I'd like to thank all the people who made this project possible, including my previous mentor, Dr. Ronald Pino, and Dr. Alan Wang at MD Anderson Cancer Center. I also want to thank my current lab members and also my wonderful collaborators at MD Anderson, as well as Notre Dame and Indiana University. I want to thank all the funding agencies that trust my research, and then together we can make cancer research better. We can eventually help a lot of cancer patients.
Thank you, Dr. Liu, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box. Dr. Liu, as well as biotechnology scientist Anna Vervoort, will answer as many questions as time permits. And our first question is for you, Dr. Shin. How do you calculate how a biosimilar binds to VEGF?
Hi. So to calculate a biosimilar binding to VEGF, I think this is a biophysical question. So I'm assuming the audience is talking about a pure compound binding to a purified VEGF recombinant protein, for instance. In this case, I personally don't have too much experience on this assay, but I think something like a surface plasmon resonance assays using Biaco from GE is a good solution to this. So, but I really want to direct this to like some expert who has more experience in biophysical binding assays, which I do not have. So, yeah, thank you.
Okay. Have you tested other PI3K isoforms, alpha, beta, delta, gamma, selective inhibitors?
Yeah, that's a good question.
Please answer that part, and then I'll give you the second part.
Yes. So that's a good question. You see that towards the end of the presentation, we did test the two PI3K isoform inhibitors, and both of them did show impressive combination effect when combined with immunotherapy. So there are also two back-to-back Nature papers published later last year suggesting PI3 kinase gamma inhibitors also can inhibit myeloid cells and then synergize well with immunotherapy. So it's kind of in line with our reports. So that's kind of answering the first question. Yes, we did. Other people also did. The selective inhibitors are believed to be better than these pan inhibitors because they are more selective and more specific.
And so the second part of this same question is, isn't a pan PI3K inhibitor toxic to T cell survival?
So the inhibitor we use BEZ235 at high concentration is toxic to T cells, but as we actually select this particular one because based on a paper, a Flick and all, that paper earlier published than us showing that this inhibitor is generally tolerated by T cells. So that's why we chose this one. So we don't entirely know why this inhibitor is not inhibiting T cells as one would expect, but it has low toxicity to T cells. But that's a good question because that is a concern. Using high dose of PI3K inhibitor would affect the T cell activity. That's why I think when you design these kind of combinations, it's important to reach like a great window of dosing, and then so that it can inhibit MDSC enough, while at the same time largely preserve activity of T cells. So if you use a very high dose for the drug, yes, I think in principle you will inhibit T cells altogether.
How do you define development of castrate resistance in the CPP SML mouse?
Right. So I'm sorry I didn't get too much time to characterize this part in the presentation. So basically, for this model, we did this combination treatment. First, we did castration, surgical castration, remove the testicle of the mice. So there is a very little residual, like supply of androgen through the body of the mice. At the same time, we use enzalutamide, which is the second generation androgen receptor inhibitor, to totally shut down the AR signaling. So by doing this together, we believe we remove all the signalings from androgen and then mimic the androgen deprivation therapy in the clinic. And then once we do that in this mouse model, in the first week, when we do imaging following up the tumor volume or bioluminescence imaging following up the tumors, the signals from the tumors, you will see a drop. We saw a drop of the signals. We saw a drop of the tumor volume, but the tumors quickly catch up in 3 weeks after, based on imaging results. And then we think that this is again consistent with a lot of clinical observations, as well as a few other mouse models that's based on PTEN deletion. There's an immediate effect from ADT in the mouse models as well as in the clinic. The problem is this emergence of the resistance, and then we define this because we saw this relapse of the kind of a regrow of the tumor bigger than the original size before the surgical castration and enzalutamide treatment. So that's how we define the CRPC. And then again, here I just want to stress that we define this based on the primary tumor. We don't have the resolution to detect the metastasis to call whether this is a CRPC or not. So the fact that they do exist, these metastases, they do develop after all this treatment, suggesting they are resistant to such androgen supply removal and then cutting. So we believe they are CRPC. They can be defined as metastatic CRPC, but we don't have a tracking for the metastasis through the whole process. For metastasis, we are looking at endpoint. For primary tumor, we do have data to track the process and then know there is a involvement and then an emergence of the CRPC. Yeah, thank you.
Have you looked at combinations with PD1 or CTLA4 separately and not just immune checkpoint blockade? And what was the rationale for using immune checkpoint blockade?
That's a good question. So in the paper, we did not separate PD1 and CTLA4 treatment. We used them together in a cocktail. One of the reasons is because that CTLA4 alone was not effective for the clinical use in the advanced prostate cancer based on reports, and then that's why we kind of want to use this combination to start with, and then surprisingly we did see even the combination didn't work well. That's why we kind of consistently use this combination of PD1 and CTLA4 antibodies as ICB used in the paper, and then later we did separate them. We particularly test the PD1 antibodies, which is a kind of more popular for immunotherapy treatment nowadays. So we use the PD1 alone in combination with the MDSC targeting agent, and we also saw a similar level of this synergistic effect. We didn't include that in the paper, but because those were done later after the paper was accepted. But we did separate them, and then PD1 antibody alone also have pretty strong effect when used together with MDSC targeting drugs.
Why is the MDSC effect minimal when you have a strong effect in proliferation assay, cabozantinib, and BEZ?
Why is I'm not entirely sure what this question is referring to, but I can answer to my best. It's I think one level to understand is we did this assay to measure the sensitivity of the main cell populations we care most from the tumor, the cancer cells, MDSCs, and T cells, and measure their response respectively to either the cabo treatment or BZ treatment in a dose-dependent manner in vitro. That's what we did. And we saw that MDSCs are most sensitive. You can, at certain concentrations that you already kill all the MDSCs, while the cancer cells or T cells are still alive. So that's kind of the basic observation we had, and then that's what we reported. And then at this moment, we don't entirely sure why MDSCs are most sensitive, because there are so many answers, so many possibilities why one cell type is more sensitive to one drug compared to other cell types. Potentially is probably the most straightforward possible answer is that the MDSCs rely on the pathways that are inhibited by those drugs more than other cells like cancer cells or T cells that we tested. So, but again, this answer is kind of philosophical answer, is not a mechanistic answer. So we are in the process of categorizing deeper why MDSCs are sensitive to certain PI3K inhibitors compared to other cell types right now, and then hopefully we can come back with better answers in the near future.
Great. And the next one is for you and for Anna, and it's a three-parter. So how about if I read the whole thing, and then you can, let me know if you need me to read, question 2 and question 3 again. One of the knockout in the chimeric mice mentioned was SMAD4. Does TGF-beta play a role in development of MDSC, and why was TGF-beta not measured in the cytokine panel? So again, the first part is one of the knockout in the chimeric mice mentioned was SMAD4. Does TGF-beta play a role in the development of MDSC?
Yeah, so that's a good question. So, so the mouse model we have used here have SMAD4 in conjunction with P10 and PV3 loss of function all in the mouse prostate specifically, not in other cells or tissues. So that means we do not perturb the SMAD signaling in myeloid cells, including MDSCs. So we did not delete SMAD4 in those cells. So those pathways, if important, are still intact in MDSCs. So far, I think that the reports on TGF-beta signaling or BMP signaling through SMAD pathways on MDSC differentiation of functions is limited. But that's a good question. I think that's definitely a good thing to test, because TGF-beta signaling seems to be pretty heavily strong immunosuppressive activity in various aspects on T-cell immunity. If we found that T-cell TGF-beta signaling is also involved in MDSC, that would be very interesting and kind of provide more insight how we can design better immunotherapeutics based on targeting TGF-beta signaling. That's a very good question, a very good direction to explore further. So the second part of the question, why TGF-beta is not measured in the cytokine array? I don't think a TGF-beta as a cytokine is included in the cytokine array panel. But apparently, if it should be included if the reagents will be available soon. So it should be included because it's so important.
And Anna, would you like to weigh on this one too, and let me know if you need me to read any part of it over again?
No, the only part I would say is that the reason why TGF-beta wasn't measured on the cytokine panel is because it's not included in the cytokine panel, most likely because there was a cross-reactivity issue with other content. So you would have to follow up with a different method, either traditional Western or Q-kits or something like that.
Okay, and next one is for you, Dr. Shen. In your view, which PI3K isoform is driving MDSC inhibition, and is the P10 deficiency dependent? Is this P10 deficiency dependent?
So for that question, which PI3K isoforms? As I said, there were 2 back-to-back papers reporting PI3K gamma is essential for MDSC or MDSC-like immunosuppressive myeloid cells in different mouse models. So and those 2 papers, one of them, I think at least one of them is based on knockout study, so it's pretty definitive addressing this question. And then I don't necessarily know whether other isoforms, rather besides gamma, is also essential. Based on our data using the inhibitors, it seems to be also very important, but the inhibitors are generally not as definitive as providing genetic evidence, so we don't have genetic evidence to knock out different isoforms of PI3K in the MDSC lineage to answer this question definitively. So I think this is something that we or others in the field should do, just to specifically knock out PI3K inhibitors, PI3K isoforms in the MDSC lineage, and then test whether which isoform is most important, or maybe they work together, or maybe one is more important than others. But the question is how to do it, because so far there's no MDSC specific cree or something you can use to generate a conditional knockout in this lineage. So it's something still some technical challenge there, but I think it's something that people should work on, including us. And then second part of the question is sorry, what's the second part of the question?
Do you think there are differences?
Of the isoforms?
Oh, I'm sorry. I moved that question over. Hang on one second.
Oh.
In your view, which PI3K isoform is driving MDSC inhibition, and is this PTEN deficiency dependent?
PTEN, the second part is the PTEN deficient dependent. Also, again, so in the mouse model, the PTEN is on the cancer cells, not on the MDSCs. So I think in the MDSCs, as we showed, the PI3K signaling is important, and then whether that's driven by PTEN loss of function, apparently unlikely to be genetically caused by that, because MDSCs, I don't think they have a PTEN deletions in them, no matter it's in the experimental system or in clinical samples. So, but again, PTEN can be regulated by many different levels, including silencing of the transcription. Do MDSC have a lower level of PTEN compared to normal myeloid cells? Possibly. We didn't test that, but that's a good question. I think if we test that and it turns out to be true, it could explain partly why PI3K signaling is hyperactivity in MDSCs.
Which cells do you use, like MDSC, in your assay? Human, mouse, and do you think there are differences?
Yeah. So in our assays, and I believe most of the assays published in papers, people use mouse MDSCs. These MDSCs can include CD11b GR1 positive cells or their subsets isolated from either the spleen or the tumor itself from mouse models of cancer. And then for us, we isolate MDSCs from tumors using Myeloid kits. And then we so far we have not used the MDSC assay from humans because in humans it's still controversial what's the best markers to use to identify MDSCs. And then there are papers that have done that to isolate MDSC-like cells from humans, and then they do show that those cells also suppress T cells. So you can search those papers and then see how they do it. So there are a combination of markers like CD33, like CD45 high or CD14 high or CD15 high, depending on which type of population you use. And also all those cells have HLA DR low, suggesting they have low MHC class 2 mechanism to present antigens. That's not their main function, I guess. So, but they have a specific markers they use. But again, the field is still debating what's the best marker to use to isolate human MDSCs. So I'm sure there are the differences between human and mouse MDSCs if you look at the molecular details. But again, so remember, we define MDSC using functional assays. If you isolate the population of myeloid cells during tumor development that can suppress T cells, you can call it MDSCs. If on that level, I think they are the same, right? Functionally, they are the same, and overall speaking. But once you look into deeper mechanisms, and particularly what genes are expressed in each population, which pathways are enriched, I'm sure you will see a lot of differences.
And it looks like we have time for one more question. Is MDSC infiltrate related to PTEN loss in CRPC similar to how others have shown for TILs and PTEN in melanoma?
So MDSC infiltrate in CRPC. So there's a recent paper published from a City of Hope, a group from City of Hope, showed that MDSCs are correlated, an increase of MDSC infiltration is correlated with a CRPC development. And then so far there are not too many papers studying MDSC infiltration in prostate cancer yet. That's one paper, and us, we look at actually as we report in the paper, our samples are not CRPC. The human samples are not CRPC. Those are primary, untreated, treatment naive human prostate tumor samples. So we don't have data to know how much MDSC infiltration is in MDSC, and we don't know either whether it's correlated with PTEN deletion. And also, I'm sorry, I don't have data to compare that with TILs or PTEN status in melanoma. So in melanoma, there are reports to show MDSC also infiltrate. But again, melanoma generally respond well to immunotherapy, probably thanks to their high immunogenicity. So for melanoma, MDSC may not be as potent as in prostate cancer. But that's just my speculation. I don't have data to back up that.
Well, thank you. I would like to once again thank Dr. Liu for his presentation. Do you have any final comments?
Yeah, so I really appreciate this opportunity to present our study. And then, as I said, we are in my new lab at Notre Dame. We are in the process of categorizing several different fronts of MDSC and further understand how they suppress T cells. And one particular thing is that we look at it and then recently found that MDSC can induce like a lot of protein post-translational modifications in T cells, in particular protein nitrations in T cells. So we are trying to finish this story and then try to report to the field. And then I think MDSCs are certainly very important for cancer immunotherapy, and more people realize this. And then there are a lot of interest in developing therapeutics against MDSC as well in the pharmaceutical company. So using like protein profilers are one of the best tools to study those because they are so heavily rely on cytokines for their functions. So yeah, again, thank you so much for the opportunity, and I would love to participate in similar opportunities in the near future.
Well, thank you again very much, and I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, Bio-Techne, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE tab at the top right and follow the process to receive your credits. This webcast can be viewed on demand through July 2018. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.